right. Don't confuse NADH and NADPH, though. This is the light reaction. You can see how it's requiring those photons, how it's requiring that light energy in order for all of this to work. Any questions on it? Okay. Now, what we're going to do is start the last part of photosynthesis, which is the dark reaction, or the Calvin cycle, or the light-independent reaction. However, whatever you want to call it, it's the second part, or of cellular or photosynthesis. The Calvin cycle, this dark reaction, requires the chemical energy that's been produced, requires all of this ATP and NADPH the light reaction has generated. The Calvin cycle also requires carbon dioxide. Carbon dioxide. So what we're going to see here, like I did earlier, we're going to have ATP put in all of its energy to become ADP and that inorganic phosphate again. We're going to see how NADP plus, or NADPH is oxidized back to NADP plus. And we're going to see how carbon dioxide is going to be reduced to carbohydrates. You can actually do like this to, however many it's reducing. So carbon dioxide becoming our carbohydrate. This is what's happening in the Calvin cycle. It's light independent, dark reaction. Does not require any sunlight. Does, however, require the chemical energy that we have made from the sunlight. So let's look at it real quick. The way your book has it broken down is they're showing 3 carbon dioxides coming in at one time. Because once again, we're going to have to count those carbons. We're going to, how we did glycolysis, we split it up into 2, 3 carbons, and then through the Krebs cycle, we broke it down completely, like by releasing all those carbon dioxides. Well, now what we're going to do is we're going to see 1 CO2, 2 CO2s, 3 CO2s come in through this Calvin cycle to generate that sugar. Okay? So when we're going through this, imagine this circle happening 3 times to get to our one product. Does that make sense? So we've got these 3 carbon dioxides. The first step of the Calvin cycle is known as the carbon fixation. We have to fix this CO2, this inorganic molecule, in an organic molecule. So the beginning of the Calvin cycle is known as the carbon fixation. We have a 5-carbon sugar. This 5-carbon sugar is known as ribulose bisphosphate. Ribulose bisphosphate. RUBP is an abbreviation for it. It might be a little easier to remember. RUBP is a 5-carbon sugar. RUBP is going to combine with that carbon dioxide, that 1 carbon. We're going to take this 5-carbon RUBP, combine it with this 1 carbon, and now I've got a 6-carbon molecule, right? The 6-carbon molecule, it's known as Rubisco. But the thing is, it's a very, very unstable carbon molecule, this 6-carbon molecule. So almost immediately in this carbon fixation stage, we bring the CO2 in. Almost immediately, we're going to see this molecule split apart and create 2 3-carbon sugars. Everybody see that? We have RUBP, a 5-carbon sugar. This 5-carbon sugar comes in contact with that 1 CO2 from the atmosphere, carbon dioxide, that we exhale as a waste product. It's a waste product to us. The plants want it. There's that one carbon dioxide. It comes in. Forms a 6-carbon, let me put it like this, forms a 6-carbon sugar now known as rubisco. This rubisco, I'm sorry? Well, I'm just showing one at the moment, but there are 3. This rubisco is very unstable. Doesn't like to kind of be here, so it'll split almost immediately into 2 3-carbon sugars. These 3-carbon sugars are 3-phosphoglycerate because we've got this phosphate on both sides. Okay, here's my 5-carbon sugar. Imagine phosphates on either side. In comes the CO2, splits immediately. And then it has like that phosphate on it. But it splits immediately. 2 3-carbon sugars. Now, we are bringing 3 CO2s in at a time. So here we're going to do it once and split. We're going to do it again and split. And we're going to do it a 3rd time and split. So in actuality, how many 3-carbon molecules have we ended up with? We've ended up with 6 because we split it 3 times. Does that make sense? Sarah?
Is that the name of the
3-phosphoglycerate is the 3-carbon sugar. So we're going to do this 3 times. 5 carbon and 1 carbon come together and splitting. Gives me 2, happens again. Gives me 4, happens again, gives me 6. I've got 6 now, 6 3-carbon sugars, or 6 3-phosphoglycerates. Any questions on that? Because you got to get it all to understand, to keep going. You got to understand this part to keep going. RUBP. So we had 15 carbons here, actually, to start with, right? With 3 5-carbon molecules, that gives us 15 carbons. We add 3 more to it. How many does that give us? 3 plus 15? 18 carbons. Now we're going to split those in half. How many, if we've got 6 3-carbon molecules, how many carbons do we have? We still have 18 carbons. Okay, we brought 3 carbons in, but we haven't lost any yet. So we're back up to the 18. We're still at 18. This is the carbon fixation stage. How we bring carbon dioxide into an organic molecule. The next phase of the Calvin cycle is how or when we are going to reduce that CO2. Carbon dioxide has to be reduced to make a sugar. Reduction, gain of electrons. It's going to pick up these hydrogens and these electrons and become reduced. This happens in this next step. We have this 3-phosphoglycerate. There are 6 of them. So 6 of them require 6 ATP molecules. In comes the ATPs. We're going to put a phosphate on the other side. We have now formed a phosphate on both sides. We have 1,3-phosphoglycerate. It's just a 3-carbon sugar. We've attached that phosphate group. We've given it our energy. We've now created a phosphate on both sides. You see how it was only on this side here? Now we've got one on this side, a 1,3-bisphosphoglycerate. ATP comes in, donates its phosphate group, phosphorylates the 3-phosphoglycerate to 1,3-phosphoglycerate, bisphosphoglycerate. Bis meaning 2 phosphates. 1,3-bisphosphoglycerate. Okay?
So you have 6 of the 1, 3, 5.
Bis, exactly. We have 6 1,3-bisphosphoglycerate. We had 6 3-carbon sugars here. In comes ATP. We still have just 6 3-carbon sugars. We still have 18 carbons altogether. Okay? Now we're going to see that reduction. NADPH is going to become oxidized back to NADP+. NADPH is going to be oxidized to NADP+. What's it going to do? It's going to give these 2 hydrogens and their electrons to that 1,3-bisphosphoglycerate. The phosphate from the ATP that went on to generate the energy that allowed this reaction to happen because we had to have an input of energy, input of the NADPH energy. We got this reaction going. Off comes that inorganic phosphate. And now we have 6 3-carbon molecules that are now reduced to 3 glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Have you heard of that molecule before? Yeah, where? Where'd you hear of G3P? Glyceraldehyde 3-phosphate. Not in the Krebs cycle. In glycolysis. In glycolysis. I'm going to pull up this overhead of glycolysis. Now, glycolysis, we took glucose and we reduced it down to pyruvate, right? Brought it all the way down to pyruvate, and then the pyruvate, oxidized it to pyruvate. And then the pyruvate was oxidized to carbon dioxide. In the Calvin cycle, we are taking carbon dioxide and trying to reduce it. Well, we have G3P, 1,3-bisphosphoglycerate, 3-phosphoglycerate. Here's this picture that you've probably seen before. We had the pyruvate down here. Well, let's come up a little bit. 3-phosphoglycerate, 1,3-bisphosphoglycerate. Did I put the other one? And at the very top, when glucose was split, what do we have? I can't get it on there. The G3P, or glyceraldehyde 3-phosphates. So in glycolysis, we were going boom, boom, boom. In the Calvin cycle, we're going boom, 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 working our way back up to this. Does that make sense? You see that? You see how that's going there? Amazing. It's not the exact reversal of reactions, is it? I'm not doing the exact opposite of what I did in glycolysis, but I'm getting the same results. I'm making the same sugars. It only makes sense. If we're trying to get to this G3P to make glucose, why wouldn't we take glucose and break it down to this G3P? Okay? Not exactly the same reversal of reactions, but the same products coming out. So now we have 6 glyceraldehyde 3-phosphates. Once we have accumulated at least 6 of these G3Ps, that means we have to do this circle 3 times to get 6 G3Ps. We have 18 carbons here. If I've got 18 carbons accumulated, I now can pull the 3 out. So I put 3 CO2s in here. I'm pulling out 1 G3P, but it's 3 carbons, isn't it? And if I'm taking one of these 6 molecules away, that leaves me with how many? Leaves me with 5 G3Ps, and 5 times 3 is 15. What did I start off with up here? 15 carbons. Okay? Yes, ma'am.
So at the end of phase 2, the products are 5 G3Ps and 1.
At the end of phase 2, we're going to end up with 5 G3Ps, and we're going to pull out one GP3, G3P. Goes into storage, exactly. So by the time we're done with this phase 2 reduction, we've created those 6 G3P molecules, those 6 glyceraldehyde 3-phosphates. Once I have 6, I can take one of them out and, like she said, put it in storage. Now I've got 5 more left that's going to go back, regenerate through a series of reactions our 5-carbon RUBP. Well, here in this 3rd phase, we're regenerating that RUBP. It requires more energy. This is where that non-cyclic or that cyclic pathway came in, because we need ATP. We need to phosphorylate this molecule again. We need to have RUBP has phosphate groups attached to it. Glyceraldehyde 3 only has 1 phosphate group. We're going to convert it all the way back to a 5-carbon molecule that has a phosphate group on either side. We need ATP to do that. So this is why the cyclic pathway has to generate more ATP. I need 6 ATPs for the 3 carbon dioxides, but I need 3 more ATPs for the reformation of RUBP. And that's where the cyclic pathway is going to make me more ATP, more energy. Okay? And our overall objective of phase 3 is simply to take this 5 3-carbon molecules through a series of enzymatic reactions, produce 3 5-carbon molecules, and start all over again. I cannot take out a 3-carbon molecule until I've put 3 carbons in. What goes in must come out. I cannot take out if I didn't put in. That's why this cycle takes 3 turns before we can take out 1 G3P, because I've got to be able to regenerate this RUBP. And I won't be able to regenerate it if I take a G3P out too soon. So realistically, this cycle to make 1 glucose molecule has to go through how many times? No? Has to go through 6 times. I have to bring in 6 CO2s, and that's my formula, right? 6 carbon dioxides gives me 1 glucose molecule. Put 6 CO2s in, gets 1 glucose out. I've got to put, this is showing 3 carbons coming in, getting 1 G3P. I got to put 3 more carbons in, get a second G3P to come together to form my glucose. This picture diagram here is representing 3 revolutions. So imagine 2 pictures with 3 revolutions or 6 revolutions. 6 CO2s have to come in to form 1 glucose molecule. But it's not just glucose that plants are storing, right? They're storing carbohydrates overall. They're building those glucoses, or they're going to build sucrose, or they're going to, build cellulose. They're going to build all kinds of different sugar molecules. But this is the way to do it. That G3P can go and become really any organic molecule, any carbohydrate overall. Any questions on the Calvin cycle? It requires ATP, it requires NADPH, and it requires that carbon dioxide. And what you get out is your sugar, your NADP plus oxidized again, and ADP. Needs more ADP than NADPH, so it has that cyclic pathway in the light reaction to make that extra ATP.
This occurs in the space.
This actually, you're right, I don't think I said that. This reaction here, this Calvin cycle, occurs in the stroma, in that fluid that surrounds the chloroplast. The light reaction occurs in the thylakoid membrane. The cyclic and non-cyclic pathways are in the thylakoid membrane, which we talked about. But the dark reaction, or the Calvin cycle, is occurring in the stroma.
So all the ATPs that are the byproducts of using the inorganic phosphate are in the stroma. Then it becomes ATP when the hydrogen ions come along the space.
Exactly. All of those ADPs, okay, so now we've generated all of our energy. We've got ADP and NADP plus as products, right? Those are what are in the stroma. You can see here how this ADP is still in the stroma and the NADP plus is still in the stroma. So when the light reaction has that input of light energy, can generate ATP through the hydrogens coming through the ATP synthase enzyme, and reduces NADPH from those electrons traveling down the electron transport chain. Is what in the thylakoid? The non-cyclic pathway is embedded within the thylakoid membrane. It's within the thylakoid membrane. The hydrogen ions build up in the thylakoid space. Okay, but the proteins are embedded inside the thylakoid membrane. Now this is a summary of everything that's going on. And this is, of course, representing the non-cyclic pathway. But here's our light reaction. In comes light energy. In comes water. Water is split through photolysis. Splits that water molecule apart. Releases oxygen as a byproduct. Those electrons then are carried from photosystem 2 down an electron transport chain to photosystem 1. Another electron transport chain to generate the NADPH, as well as the ATP through photophosphorylation. Photophosphorylation. NADPH is reduced. Now this is our chemical energy. We have taken the sun's energy, light energy, and have converted it to chemical energy, ATP and NADPH. These are the two reactants feeding into the Calvin cycle along with our carbon dioxide. Through this cycle, we're going to produce all of those starches, carbohydrates, amino acids, fatty acids, all of our organic molecules. Basically, all of our organic molecules, everything that makes you and me, has to come initially from the plants. We cannot do it. We get it from the meats we eat. We get it from the vegetables and the fruits and things that we eat. But even those meats ate, the chicken and the cow and the pigs, and they eat the vegetables too. They eat the wheats. They eat those fruits. They have to eat it to get it into their system. We have to eat it to get it into ours. All of our organic molecules. That is how we are an organic organism, is because of the plants. They will do just fine without us. But water world would never exist. We could not survive without the plants. We would not be able to survive without the plants. But the plants would do just fine without us. So then we go into this Calvin cycle, CO2, all the chemical energy. Out comes our organic molecules and our byproducts, NADP plus and ADP and inorganic phosphate, to feed back into the light reaction to start it all over again. Yeah. Did you catch that? The algae produce enough oxygen probably for the fish. I mean, for a while, but you can't just live off of fish. You have to have the fruits and the vegetables and, some of those carbohydrates. The fish just aren't going to do it, and the kelp and stuff. The ocean produces all the bulk of the oxygen anyway, but you can't drink the water. It's all salt water, and I just don't think it would work. Okay? This is a nice little diagram to study. That chart that I gave you, that summary chart that I gave you, it was a difficult chart to come up with, but going through this, it'll help you fill out the different spots and stuff that need to be filled out. Any questions on the light reaction or the dark reaction of photosynthesis? Yes, that chart. And the exercises on the back are also very helpful. Okay. You have a chart. I don't have an extra one with me, but now the last couple pages of your text talks about different types of Calvin cycles or manipulations of getting the carbon dioxide into the plant cell and things like that. I'm not going to go into them. You can read over it, but they talk about what we call C4 plants and CAM plants. And basically, there are different ways of going through this overall Calvin cycle, incorporating different molecules. Plants can go through what's called photorespiration, where they use the light energy and utilize this oxygen instead of as a waste product, utilize it to build their own, sugars. And by going through photorespiration, it's like those desert plants that don't want to open up to release the oxygen and bring CO2 in. Some of that oxygen will build up. They'll utilize that oxygen that way. One of the cycles involves using that oxaloacetate that we saw in the Krebs cycle, okay, and incorporate that 4-carbon molecule to get into the 5-carbon of RUBP and so on and so forth. You can read over it, but I'm not going to quiz you on it, so if you don't have to worry about testing on it. Okay? Any questions on photosynthesis? That is the end of our material for exam 3. Our exam 3 is next week. Okay? Your take-home quiz is due tomorrow. Now that you have all of this, this was basically questions 2 and 3, and then 4, or 3 and 4 was dealing with photosynthesis, and 5 dealt with both respiration and photosynthesis.